The overall goal of this procedure is to generate an array of up to 252 distinctly labeled fluorescent target cells for immune assays using multi-parameter flow cytometry. This is accomplished by first labeling target lymphocytes with unique combinations and intensities of vital dyes, such as CFSE and CTV. The next step is to pulse each distinct target lymphocyte population with candidate MHC class I and MHC class II binding peptide epitopes. This is followed by extensively washing the cells to maintain the specificity of the epitopes, and then pooling the cells and injecting them into an animal. The final steps are to harvest the fluorescent tag array cells from specific tissues, label them, and assess for MHC peptide-specific responses. Ultimately, flow cytometry results can show CTL responses by measuring target cell death, and they can show CD4-positive T helper responses by measuring B-cell target upregulation of activation markers.
Well, the major advantage of this technique over conventional in vivo CTL assays is that it allows a simultaneous measurement of over 250 target cells in one animal. Demonstrating the procedure will be Ben Quah, a research fellow in my laboratory.
The procedure requires lymphocytes collected from the isolated spleen and/or lymph nodes of mice, filtered and resuspended at up to 200 million cells per milliliter in 11.5 milliliters of rPMI containing 5% fetal calf serum. Preparing the